Hello folks, Anna here again, and we're going to just keep on plugging through our neurophysiology chapter. We're going to do part 10, where we're going to talk about excitatory postsynaptic potentials and inhibitory postsynaptic potentials. All right, so let's kind of organize our thoughts here. The first thing I really want you to think about is the fact that these things can be excitatory, excitatory or inhibitory, okay? And what this means is excitatory starts depolarization, okay? Whereas inhibitory slows, stops, hampers depolarization, okay? The point of an inhibitory stimulus is basically to hyperpolarize a membrane, making it harder for anything to happen, okay? Now, when we are organizing our thoughts, remember you've got local potentials versus action potentials. All right. Something to keep in mind is action potentials are always excitatory, okay? It is local potentials that can excite or, my handwriting, inhibit. Okay, so keep those thoughts in mind as well. Okay, now when we're dealing with these, okay, as we continue to organize our thoughts, you have what we call excitatory postsynaptic. Actually, I'm going to post, and I'm going to capitalize that S for a reason. Postsynaptic potentials, okay? And we're going to abbreviate that EPSP. So you've got excitatory postsynaptic potential, okay? But you can, what happened here? Did it just twist on me? Oh, that's interesting. Oh, how fun. Okay, you can also have inhibitory postsynaptic potentials. All right, and we're going to abbreviate that IPSP, okay, for inhibitory postsynaptic potential, okay? Now, this may seem a little obvious, but you need to have a gate that accepts an excitatory chemical, okay, an excitatory neurotransmitter. And over here, you need to have a gate that responds to an inhibitory chemical or neurotransmitter. What's interesting is when you think about how, whoops, that's not what I wanted to happen. I wanted to do this. When you think about this, you can actually have the same chemical excite or inhibit. What it does depends on the target organ. Okay, so different target organs will have receptors or gates that respond to the same chemical in different ways, which is kind of cool when you really think about it, okay? And again, what you want to really focus on right here is that the point of an excitatory is to get a local potential that will eventually lead to an action potential. Whereas with inhibitory, the point is to stop communication, okay? And there are good reasons to do that. It's not just that your cells are being difficult, all right? There are good reasons to want to reduce the possibility of having a local potential that leads to a action potential, okay? Let's look at some pictures now. All right, for some reason I've got some random lines at the top here and I don't know why, so we're just going to pretend that they don't, oh, they were supposed to be arrows. That's what they were supposed to be. But they did not come over with the PDF conversion into this program. So anyway, ignore these, all right? What I want you to focus on is I've got over on this side a graded potential example and over here an action potential. Now, the other thing to focus on is on the graded potentials, you're dealing with either a chemical gate or mechanical gate. And so if you look at those, you can see there's no little doors shutting the gate. Whereas if you look over here, you're dealing with voltage gates. And you can see their little arms here, and the inactivation gate right there that's going to block movement. I'm going to erase this before it gets stuck. Okay? So let's keep our picture looking nice. So the initial stimulus is going to use a chemical right here, a neurotransmitter, to open the gate. And so in this case, you see that sodium is going to go in and potassium is going to go out. It doesn't have to be sodium and potassium. So you can see over here with this chemical gate, it's working with potassium. But when I put this neurotransmitter in here, what I want you to focus on is these chloride ions that are going in. Okay? Now, what kind of gate it is determines what's going to happen. So in this example, they're using this one to show depolarization. So you've got resting membrane potential, depolarizes, comes back down, didn't reach threshold. Okay? Here, we're hyperpolarizing the membrane. So when these chloride ions come in and these potassium ions go out, it makes the inside more negative. So you can see right here, it's going like this, and then it dips, hyperpolarizes, and then comes back up to resting membrane potential. Okay? Now the action potential, again, controlled by voltage gates. Sodium goes in, you're right here, reaches threshold, generates action potential, and then it repolarizes and goes back to resting membrane potential. Okay? So this is a nice little comparison of an excitatory postsynaptic potential and an excitatory, oh my gosh, that's such a mess. Hold on. All right, and so these are good examples of what an excitatory postsynaptic potential does there, and then what an inhibitory postsynaptic potential does right there. Okay, let's look at the next picture. All right, so again, we got some import issues. So these are little boxes, little arrows. These little boxes are arrows. Okay, and what we're showing over here is the excitatory postsynaptic potential versus the inhibitory postsynaptic potential. And you can see this one is hyperpolarizing and this one is depolarizing. So you come in here using a chemical, a neurotransmitter that opens up a gate that allows sodium to come in, making the inside less negative, so it depolarizes. It doesn't reach threshold, so it's going to fizzle out, and it's just going to be a local potential in this case. And then it's going to repolarize, and you're going to go back to resting membrane potential. Over on the second picture, you're dealing with resting membrane potential, but this one is going to open gates for, let's say, chloride ions. The chloride ions are going to come in, and they're going to hyperpolarize. So right here, I'll just circle it, hyperpolarize the membrane, and then resting membrane potential is going to go back up to normal. Okay, so this one is inhibiting. Okay, in both cases, both of these are subthreshold signals. Now obviously The IPSP is going to be subthreshold because it's hyperpolarizing. But this one is also a subthreshold signal that did not result in an action potential. All right, let's move over and do some more whiteboard or blackboard definitions. All right, so one of the concepts to remember is when you have a subthreshold signal, you're going to get at the most local potentials. And these local potentials could depolarize or hyperpolarize. But you're not going to get an action potential because it's subthreshold, so no action potential is possible because you have to reach threshold in order to open up voltage gates, okay? But what you can do to counter this is called summation. So summation is where you take several, I'm going to abbreviate this sub-T signals, so subthreshold signals, and add them together to get a bigger impact. Now we looked at summation with the muscle physiology chapter as well. We looked at wave summation and we looked at temporal summation. This is the same basic idea, but in this case you are adding together excitatory postsynaptic potentials and inhibitory inhibitory postsynaptic potentials, okay? And it doesn't have to be all excitatory or all inhibitory. You could do like 2 excitatory and 1 inhibitory, or 10 inhibitory and 1 excitatory, okay? Now, if you remember with summation, we had 2 basic types that we could deal with. We could deal with wave summation. Sorry, that's not what I meant to write. I'm thinking about the wrong thing. We can deal with spatial summation, or we can deal with temporal summation. So spatial summation is when you have 2 plus presynaptic neurons near each other at the synapse, okay? Temporal summation is where you have, we'll just use this to simplify it, 1 neuron firing rapidly, okay? So you decrease the amount of time between submissions. So you decrease time between stimuli, okay, from 1 neuron. And then you add them together and you can potentially reach an action potential. All right, so let's look at this on a picture. All right, so this is from an old book, but I really like what they've done here because they've got an E and you've got a green neuron right here. This is an excitatory neuron. And so you've got an E, you got E2 and E1, you've got E, but then right here you can see this one's red and this one is inhibitory. Okay, so this is my postsynaptic neuron and that is the presynaptic neuron. Okay, then this whole thing right here goes with this particular graph. Okay, so you've got -70 resting membrane potential right here. I've got this neuron sending a potential down, sending an action potential down, releases neurotransmitters, synapses with the cell body, opens up chemical gates, and it's going to cause depolarization. It is still subthreshold, so it doesn't reach -55, and it goes back to resting membrane potential. And then it shows that it does a second stimulus, which is also subthreshold, and then it goes back to resting membrane potential. Okay, so this is subthreshold stimulus with no summation. Now on B, we're going to show you temporal summation. So you can see the structures are the same. All right, it's the same presynaptic neuron, the same postsynaptic neuron. What is different is how frequently the presynaptic neuron is firing. So I have my resting membrane potential and I send a signal. It is subthreshold. It begins to repolarize the membrane. However, it doesn't get all the way back down to here before a second excitatory signal comes in. Now, this opens up those gates and it allows more sodium to come in, enough that it can add together the sodium from the previous signal. That sodium hasn't all been pumped out. So now it reaches threshold and it generates an action potential, comes back down, repolarizes. Okay? Now in the third box, we're going to look at spatial summation. So I have presynaptic neuron 1 and presynaptic neuron 2. They both send a signal at the exact same time because I have two chemical gates open. Enough sodium comes in to reach threshold and I generate an action potential. In the final box, I have a presynaptic neuron that is using a neurotransmitter that excites, and I have a presynaptic neuron that is using a neurotransmitter that inhibits. Okay? So I've got resting membrane potential. The inhibitory one is firing, and that causes that membrane to hyperpolarize. It begins to drift back up. Now I'm going to have my excitatory and my inhibitory both fire at the same time while the resting membrane potential is at -70. If we come back here, I want you to look at the height. All right, that is the height if I just had an excitatory signal. Not enough to reach threshold, but pretty darn close. But when I look at this with the inhibitory, this basically cancels out half of the effect of that excitatory signal. Okay? So that's a nice example of how inhibitory can reduce the impact of an excitatory signal. Okay? All right, so that was looking at your chemical synapses and how they work together with excitatory postsynaptic potentials and inhibitory postsynaptic potentials. That's it for the slides for this section. I'm going to go ahead and stop the recording.